Hello and welcome everybody. My name is Daniel Yoshikawa, and we'll be starting this right now. I am Bio-Rad's North American Product Manager for Proteomics. I am your moderator of today's webinar. Thank you all for attending today's free webinar sponsored by Bio-Rad. This presentation will be recorded, and you will receive the playback link by email following the presentation. We will discuss protein purification using the new Profinia automated Affinity-Tagged protein purification system. At the end of the presentation, we will have a question and answer session. Questions are submitted using the text chat feature, which some of you have already been using. It's located at the top of your screen. Although we will not respond to questions until the end, you are welcome to submit questions anytime throughout the presenter's presentation. Should you experience any technical difficulties, please send us a message using the text chat. Address the message to CK. CK is our technical specialist. We are pleased to have Dr. Laurie Usinger speak to us about the Profinia system. Laurie is as the Profinia program manager and lead scientist. Laurie successfully led the development design of the consumables line supporting the Profinia system. Prior to her work on Profinia, she played key roles as project manager and lead scientist in the development of several other technologies for both electrophoresis and chromatography here at Bio-Rad. Laurie earned a BS degree in biology from the University of California at Berkeley, a PhD degree in cell biology from the University of California at Davis, and she performed her postdoctoral research at the University of California at Berkeley before joining Bio-Rad as an R&D scientist. Laurie will describe the Profinia system's technology and how it will simplify your affinity purification experience. Please welcome Dr. Laurie Usinger.
All right, well, thank you, Daniel, and thanks for logging in to our webinar seminar. So I'll just jump right in and introduce you to the Profinia protein purification system. And this is Bio-Rad's really new and what we think novel approach to affinity purification of tagged proteins. So I'd like to go over an overview or just a brief outline of the presentation. I'll talk to you a little bit about affinity-tagged purification and the methodologies. I'll introduce you to the Profinia protein purification system. We'll talk about some of the considerations of sample prep. I'll present some representative IMAC and GST purifications that we've done in-house here at Bio-Rad, and then we'll talk about parameters and variables that affect both your IMAC and GST and other types of affinity purifications. We'll then do a comparison of Profinia purifications to other methodologies that are used to purify affinity-tagged proteins. We have some comparative data that compares resin analysis from both Bio-Rad and some other resins that are on the market. And then most importantly, we'll give you some real-world applications where Profinia is being used in customers' labs. So that's a overview of the presentation. If you think about current techniques and molecular biology labs, the typical workflow is that you identify a gene of interest using some sort of molecular biology technique, such as RNAi or qPCR. Okay, there we go. Get the animation up and running. That gene is then expressed, and the ultimate goal is really to identify the protein, to define the protein's function or structure. And typically during the expression, an affinity tag is put onto your gene of interest so that you can easily purify your target from your complex life state. So when you think about affinity purification, what are some of the challenges of affinity purification? Well, in general, it's a fairly straightforward chromatographic technique, but there are some inherent challenges to affinity purification. There's a lot of hands-on time if you're using, say, gravity chromatography. If you're using a typical low-pressure chromatography system, there's a lot of setup and programming time required to get your instrument up and running. There's also, after you do the purification, you typically want to do a buffer exchange and get your protein of interest into the desired buffer. So there's a lot of time in terms of buffer prep, dialysis, etc. And ideally for affinity chromatography, the researcher just wants a system that they can use that's scalable, that's reproducible, it's automated. But with the important aspect of freeing up time so that you can actually do your research and not just spend it on affinity purification. So here is the Profinia purification system. Some of the benefits of Profinia. There we go. It's an automated system, so it's fully automated, and it's the 2D system, and the fact that here your affinity purified peak is automatically selected and diverted onto the desalting or buffer exchange cartridge on the system. So it's a 2D system, and that really gives it a unique aspect to affinity purification. It's very efficient, so the methods are pre-programmed, so you really walk up to Profinia like you would, say, a cycler, a MyCycler, or an iCycler. You basically, punch in your method of choice, you hit go, and then you're ready to run and purify your proteins. In terms of performance, it's really reproducible. So you remove the variables of, say, gravity, where you perhaps let your bed go dry, or spin columns where maybe you spun it 3 times as opposed to 4. So Profinia is really reproducible, and multiple users can get the same results from an affinity purification. So that's kind of Profinia in a nutshell. To give you more specifics of the instrument and provide an overview, the method is directly in front of the instrument. There's 4 main methods to choose from the preset methods. We have native IMAC, nature IMAC, GST, and a desalting method. We have a variety of kits that complement each of those methods, and those kits contain all the buffers and cleaning and storage reagents required to run an affinity purification, and they are inserted into the buffer cartridge section of the instrument. The cartridges we've made and redesigned, affinity and desalting cartridges, they're Luer lock, and they insert quite easily into the Profinia instrument. The instrument can purify up to 2 samples. So the samples, typically E. coli lysates, are loaded into the sample loading region of the instrument, either 1 or 2 samples. And then the samples are fractionated and purified, and the fractions are collected in the fraction collection region of the instrument. And the or purified proteins are centrally located for ease of use in terms of going and just grabbing your purified sample out of the Profinia. The Profinia comes with a software package. The instrument's not driven by the software, but there is a reporting software package that can be purchased with the instrument. It's nice in that you can actually monitor your run in real time. Now, however, if you don't want to have a laptop or a computer sitting with your instrument, you can download your file after the run and just go back and, at the convenience of your desk, look at the reports and actually print the reports for your lab notebook. So the software is a nice feature of the instrument, nice and user-friendly. So in terms of programming the methods, again, we've tried to design this instrument really to be compatible and accessible, and give it a PCR type feel, or a MoBio type feel, and that you walk up to the UI and select your method. So in this screen, you can see a representative screen of the UI. So here we've selected native IMAC and desalting. You would then, on your UI, press Next. After you've chosen your method, the UI guides you and tells you what reagents you should install, what sample tubes you should install, where you should install your sample. It tells you to check your water for your diluent, et cetera, et cetera. So it guides you through every step of the process. And after you've basically pressed start the run, you get this nice run in progress screen where the instrument or the UI tells you what the progress of your run is. It tells you when your sample is going to be eluted. It tells you exactly what time that you can expect to have your purified protein. So you can basically walk away and come back at 11:48 and say, okay, I'm ready to do my next experiment. There's also real-time data on the UI in case you don't have the software running in real time. That gives you key run parameters such as flow rate, the OD values coming off of the UV cartridge, et cetera, et cetera. So that gives you a brief overview of the UI as you set up your method. Now, at the end of the method, This is a really nice feature of the instrument also, is that you actually get to, the instrument tells you what volume of purified protein in your collected purified fraction. It tells you how much protein is there, because the firmware actually integrates underneath the peak of the collected purified protein. So it actually does a calculation, and it tells you how much protein you have there and the protein concentration. So again, this makes it nice and user-friendly to researchers who don't want to necessarily run over to the spec and take a spec reading on their protein. They'd rather get to their downstream experiments on the purified protein, which is really, the focal point of research labs. We have a variety of buffer and purification kits that are applicable to all the main methods. And so here, this slide just tells you, quickly the formulations of the buffers. We have native and denatured IMAC kits, GST kits, and desalting kits. And then we also have, within each kit, we have cleaning and storage solutions such that your instrument lines are flushed and your cartridges are flushed for use at a later date. So in terms of the way that we design the system and the way that we design the UI, again, our goal was to make it as user-friendly as possible. And so the UI and the instrument and also the consumables really integrate quite well. So the UI tells you, how many buffer solutions you need, what the minimum volume you need. It tells you on the label of each of our bottles. It tells you exactly what port you're supposed to put your buffer in, and then also the instrument guides you as to where the placement of the buffers. So again, there's not a lot of thinking about the affinity chromatography. We're going to let you think about your downstream experiments about what your protein does. In terms of the software package, so if you do want to purify proteins on Profinia with the software package, the software gives you the nice advantage that you can actually visualize your run in real time. So it gives you analysis of the data in real time. On the software package, at the end of the run, the software will give you specifics about the run in terms of elution volume, concentration of your protein, and the yield of your protein, analogous to what you see on the UI, user interface. I'll use that acronym quite a bit throughout the talk. The purified protein that is eluted into tube 1D of the instrument, and I'll show you that later on, is actually highlighted in gray. So you know exactly what part of your chromatography, what part of your profile was actually isolated. So that's a nice feature of the software. And I just described that. And then also at the end of your run, again, if you have the software, basic, or you can actually just export the chromatogram and put that into a PowerPoint, et cetera, et cetera. So the software is just a really nice feature of the instrument. I strongly recommend getting the software with the instrument. Makes it even more user-friendly. In terms of the cartridges, so again, we totally revamped our cartridge line, and we designed these cartridges specifically for Profinia. And so we've made them such that, no more packing of columns, no more batch, et cetera, et cetera. We have 2 different cartridge sizes that we offer. We have a 1 ml cartridge that's filled with 1 ml of resin that you can easily purify up to, or actually even sometimes more than 10 mg of protein per 1 ml cartridge. Typically, when you're talking about E. coli cultures, this is about 100 ml culture volume from a high expressor. Obviously, if your protein expresses at a lower level, you need to up that culture volume. And we've optimized the method such that we have a standardized flow rate we use with our preset methods. We also have a 5 ml cartridge that's available, and basically everything is scalable by a factor of 5 mls of resin, and you typically obtain 5-fold higher yield from a 5 ml cartridge, so you can get 50 mg of protein from a 5 ml Profinia run, and everything else is scalable by a factor of 5. So what I'd like to lead into now is some of the considerations with sample prep. As with any type of chromatography, and that includes, affinity purification, that includes Profinia, really the quality of your downstream purified protein is really dependent upon your sample prep. And so one should spend a few minutes analyzing your sample prior to doing the purification. So what we recommend and it's really, standard methodologies in the literature is that you do a solubility and expression screen. And so in this slide, showing you is, and the left-hand panel shows you a protein that partitions into the insoluble fraction, and the right-hand panel shows you a protein that partitions into the soluble fraction. And what we typically do in the lab, especially when we're working with proteins for the very first time, is we then scan these gels and we use quantity 1 to actually quantitate the specific amount of protein that's in your lysate. So again, on the left, we see a protein that's insoluble, that it's expressed at about a 21% expression level. So this is a high expressor. And then the right-hand panel, we quantitate with quantity 1 that's expressed at about a 7% expression level relative to total protein on your chemosity gel. Depending upon the solubility profile and the expression level, that really guides you as to which purification procedure you should use. Or not, you should use your first step, and probably a mistake that a lot of users will do is that they'll just basically lyse their culture and not even check it on a gel. And sometimes your variability between days and lyses and sonicators will certainly play a role in the success of your affinity purification. Okay, so let's look at some real data that we've generated in the lab in terms of representative IMAC and GST purifications. Out of the box, when you receive a Profinia instrument, we have developed a starter kit in the lab, and that starter kit contains all the necessary reagents needed to basically just run through a Profinia run. The key element of that starter kit, or one of the key elements, is a lyophilized control lysate that has a GST moiety at the amino terminus and a his tag at the C terminus, so that this control can be used for either of our GST or IMAC purifications. So in this slide on the upper panel, I'll spend a few minutes going over this slide because it really presents the theme of all the slides in the subsequent panels that I'll show you. The upper panel is the chromatogram that one sees with the GST purification. The blue trace is actually the UV trace coming from the Affinity cartridge. The green trace is the UV profile coming from the desalting cartridge. So again, Profinia being a 2D system has two flow cells that monitor both your affinity purification and your desalting or your buffer exchange. The gel on the left shows representative fractions from each of the main fractionations that Profinia performs. And so our gels are typically set up with markers on the left, L for sample load, flow through, the first wash. For GST, IMAC also has a second wash, and the E diluted or purified protein. So the IMAC profile is shown on the bottom, same story, UV1 for the affinity, green for the desalting. And also what the software profile gives you are icons for tube icons. So tube 1A contains the flow through, tube 1B contains the first wash, tube 1C contains a second higher stringency, higher imidazole wash, and then tube 1D is really the target fraction of interest for customers, for users. We have also optimized the kit such that for each starter kit run, you purify approximately 3 to 4 mg of protein that elutes at approximately 1 mg per mil. So when you make a 1 to 10 dilution on your Lamley, on your page del, you load 1 microgram. So we kind of a little bit of strategy went into the starter kits. I'm getting a delay on my slide advance. There we go. Okay, so this slide just really presents some representative purifications that we've done in the lab for some proteins purified under native IMAC conditions. So just the take-home point here is that you can purify a variety of proteins, a variety of sizes. Here's a 32 kD yeast shuttling protein, Down here on the right-hand panel is the chromatogram and the corresponding gel for DHFR. So again, the two UV profiles, the highlighted peak, and then standardly the purified protein that you see. And these are very typical results for basically just any protein that you express and purify, in this case from E. coli. So in this slide, we just have a summary data table of, again, a variety of proteins that we've purified in the lab, ranging in size from 10 to 150 kV from a variety of different species, purified under both native and nature and IMAC conditions. And in terms of yield and purity, these are just shotgun approaches where we've just taken 100 ml cultures and purified them with our preset methods on Profinia, average yield of about 6 mg, average purity of 93%. So that's pretty typical of representative affinity purification or his-tagged purification that you get with Profinia. So in this slide, comparable type of results for GST. So again, the take-home point, you can purify a variety of proteins. We have the chromatogram on the left, the corresponding gel image on the right. Here, your sample load. So there's our target protein. Flow through, very little protein in the flow through and in the wash, and our nicely purified target protein. So those are just a few examples from GST proteins. So what are some variables or parameters that need to be considered when you're going to purify proteins on Profinia? Well, flow rate. So certainly flow rate will affect both the time of purification and the yield of protein that you obtain. And so with Profinia, this is an example for denaturing IMAC, the preset flow rate is 1 ml per minute. So for this specific example here, 23 kD protein, when we basically shotgun and purified it through Profinia, we obtained 7.5 mg of protein in 24 minutes time. Well, as a researcher, you may say, well, you know what, time is really important to me. I want to crank that protein through. Then you just up the flow rate. Conversely, if yield is incredibly important to you, then you just decrease the flow rate, and you can get a correspondingly increase in your yield. So typically, flow rate is inversely proportional to protein yield. What I'd also like to show you on this slide is in terms of in the gel analysis, we have the 3 different flow rates, 0.5 ml, 1 ml, and 2 ml a minute. And what you can see is as you start really upping the flow rate and cranking your protein of interest, your lysate, through the cartridges, through Profinia, you'll start to see more of your target protein in your flow through and your wash. So there's, flow rate will affect multiple variables, yield, purity, and then also protein that's lost in the flow through. So start with the preset method, and if you want to optimize it for your protein of choice, then you can just go to the custom methods, and we'll talk more about the custom methods a little bit later. Scalability. So scalability is certainly important in chromatography, and with affinity chromatography in general, the methods are quite scalable, and with Profinia, we've optimized the cartridges and the methods on the instrument or on the UI so they are scalable. And so what this slide shows you here is that we've done a 1 ml purification and a 5 ml purification. You see that the chromatograms are very similar, the gel analysis are very similar, and most importantly, the scale scales by a factor of 5. So for the 1 ml purification, we used 75 ml of culture, got about 10 mg yield. We upped it by a factor of 5, and then we obtained a yield that was 5-fold higher, which is what you'd expect with really no change in purity. So Profinia is quite scalable, and again, you don't have to think about what size column you should pour. You don't have to think about how much resin to put in your column. The cartridges are there, ready for you to use. Another key variable is the amount of sample that is loaded on a per run or on a cartridge, and what affinity chromatography is really no different than standard chromatographic separations. Typically, the more protein or the more starting sample you load, the higher your yield, and so the graph on the left shows the effect of yield as you increase column capacity or increase the load on your cartridge. But the important take-home point is that yield, your purity ultimately will be compromised if you keep loading more and more sample onto your cartridge. So there's a sweet spot for Profinity cartridges and also for affinity separations, and typically that's in the 50 to 75% capacity of your column or of your cartridge. And again, the black arrows show that as you up your protein load, bottom line is you're just you're throwing some protein away because you're seeing more protein in your flow through and in your washes, and ultimately your purity takes a little bit of a hit. The same is true for GST purification. So here's just a slightly modified way to show the data. But again, there is a sweet spot in your cartridge. Your yield will increase proportionately, but your purity eventually will be compromised. Again, as you load more and more target protein, you're also loading more and more contaminants. So as you purify proteins on Profinia, it's important to keep in mind that the cartridges typically should be loaded in the 50 to 75% capacity range. So keep that in mind. Proteolysis in cold room, and how should you purify your protein? Should you purify it at room temp or in the cold? Well, certainly temperature has a pretty can have a very dramatic effect on yield. Some proteins, if you're really concerned about proteolysis, if you want to keep your sample cold, well, you can do one of two things. You can move the entire instrument in the cold room, or you can actually use these nice little convenient cooling blocks that we provide as an accessory for Profinia so that you can keep both your crude sample and your purified sample cold. But this slide just illustrates that purification temperature will affect yield. So in the upper panel, we have a chromatogram that shows an overlay of purification that was done both at room temp, the upper peaks, and in the cold. And typically what you see is when you do purifications in the cold, and this is certainly true for affinity purification, is your binding capacity decreases because your binding kinetics slow down. And on the lower panel, I'll describe that lower panel in just a second. So when we compare yields from room temp versus cold, typically you'll have a 20 to 30% drop when you do purifications in the cold. So here's the effect on yield. That's assuming you keep the flow rate the same. Well, Profinia actually is, we've designed the programs, and the UI is really smart in that it prompts you. If you're going to purify in the cold, it automatically adjusts the flow rate so that you can actually compensate for those decreased binding kinetics and up your yield by purifying in the cold, and you just decrease your flow rate. So that's actually an option that you can select in the preset methods. So I think that's about all I wanted to say on that slide. So cartridges and columns and reuse, and how do you clean and store them? So with any type of chromatography, in general, the rule of thumb is one protein per cartridge. So if you follow that rule, we recommend reusing the cartridges up to 5 times. And so we have cleaning and storage solutions built into each kit. So again, this is the advantage of getting the purification and starter kits and buffer kits, is that you have all the reagents for the purification, but you also have all the reagents for the cleaning and storage. And so this chromatogram shows the purification run, and at the end, the methods utilize a high salt buffer followed by a weak acid to strip and clean the cartridge. So this little green peak is basically contaminants that are removed typically from the affinity peak. The desalting peak is pretty inert. The cartridge at the end is then stored in the storage buffer, and they're ready for reuse. And we recommend up to 5 times of reuse. Now, if as a researcher you feel that these conditions aren't stringent enough for your needs, you can customize those cleaning protocols. You can make up buffers of your choice, and you can use the Profinia custom methods to modify the purification to, again, meet your cleaning and sanitizing needs. But in general, we provide reagents in the kits that will keep your cartridges functioning for up to 5 runs. You need to think a little bit about your protein and what method that you want to choose for Profinia. So in general, with protein size, if you have a protein that's fairly large, so if you're getting above 100 kD and above, you want to choose a slower flow rate. Really large proteins have a much higher ratio of protein to affinity tag, and so the binding affinity is not as high. So you want to slow the flow rate down so you give the protein more time to interact with the resin. Our preset methods, again, we designed the methods so that they give you flexibility, so you can choose the slow flow on the preset method, so that you can actually the instrument will slow down the flow rate for you. You don't need to think about it. In terms of solubility, after you've done that initial solubility profile analysis, if you know that you have a protein that's in the soluble fraction, choose the native purification method, at least for IMAC proteins, and vice versa for an insoluble protein. If you know that your protein of interest can be subject to proteolysis, and so you're worried about protease sensitivity, move your Profinia into the cold room, or just use cooling block for purification. So that's kind of handy in that that you don't have to actually move and work in the cold room. And unfortunately, I didn't include a picture of the cooling block on this slide, but feel free to email later and I can send you an image. It's a nice, handy little device. Also, if you're going to go in the cold room, you slow down the flow rate. So again, the UI for preset methods will give you that option to choose, or you can also change the flow rates on the preset methods to whatever flow rate you desire. And contaminants, if you know or if you think that your lysate has more contaminants than a typical we have a standard wash or an extended wash where we just up the column volumes of your wash steps. So those are, in thinking about your protein and which variable you want to choose, those are some things to think about. All right, so in general, I just want to, reiterate that we have 2 different types of method on the Profinia instrument. We have preset methods that are used they're optimized with our buffer kits that we've used for a variety of proteins. We also have customized program methods that you can go in there and you can change flow rate, wash volume. You can put in different buffers that aren't supplied in our kit. You can also modify Profinia to purify any type of tag, any type of affinity tag. So that's all done in the customized program method mode. So I'd like to quickly just compare Profinia to other types of conventional methodologies. Obviously, there's many ways that you can purify affinity tag proteins. You can use gravity, spin column, batch. You can do low pressure chromatography on very complex instruments. But I would say all those in general are fairly time consuming and tedious. And with Profinia, basically with 10 minutes of hands-on time or less, you're ready to go and purify. So to show you some data and to compare Profinia to, say, spin column purification, here's an experiment we've just done, Profinia versus spin. And with the advantages of Profinia for comparing it to spin column purification, that is a standard technique in the field. Yield is really not changed, but purity oftentimes will be compromised in spin columns if you're spinning them at such high speed. But also hands-on time, 10 minutes for Profinia, whereas typically with spin columns you're dealing with a centrifuge for almost an hour. In terms of gravity purification, here is a slide where we've shown Profinia in comparison to gravity purifications, triplicate runs, really the same theme here, and that is of the essence and time is important for you. 10 minutes for Profinia, but really if you're dealing with gravity, you're sitting in front of some gravity columns for almost an hour. So Profinia gives you the advantage of just a big time saver. If you're comparing Profinia to some low pressure systems, and if you like plumbing chromatography instruments, compare it to an FPLC with Profinia. Here's the gel on the left and FPLC on the right. The big advantage here is Profinia gives you the 2D separation profile, so it automatically diverts your affinity peak onto the desalting peak, and you get a nice buffer exchange purified protein. With a standard chromatography system, you have to first do the affinity purification. You then have to identify the peak, which was typically fractionated in a fraction collector. You then have to pull those peaks. You then have to put those peaks back onto an instrument that was set up with new solutions, and have to isolate. Here is that your time, again, with Profinia, it's 10 minutes, whereas with the standard chromatography low pressure system, you're dealing with well over an hour of purification. Profinia also does desalting, so the follow-up to affinity purification, especially if you're doing 2 samples, is that you have to remove salt, which is a contaminant. So in IMAC purifications, you have 250 millimolar midazole. Well, there's a desalting step on Profinia where in 15 minutes you can purify, or you can separate your protein from contaminating salt. And so again, time, Profinia versus standard dialysis. Dialysis minimum of 2 hours, but typically overnight, and certainly a lot less buffer used with Profinia. So let me take you into a comparison of resins. I'll just go through this quickly. We have our Profinity media, our Profinity resin, and then 2 resins that are commercially available on the market. And what I want to show you here for IMAC is that, and here's the corresponding gel analysis. For IMAC, typically your yields are about the same, but we find in our hands we get higher purity for proteins that are purified over IMAC resin that are packaged in our Profinia cartridges, which are optimized for Profinia. Same, basically similar results for GST methodologies, and that here is a chromatographic profile from our GST resin that's packed in our Profinity cartridges, and again, 2 different resins that are available on the market. And what you find is that here's a gel analysis of our Bio-Rad resin versus 2 other agarose GST resins that are on the market. Again, weave resin and purities are pretty comparable across different resins. But again, with Profinia, we have buffer kits that are compatible, they're optimized for the instrument. We have cartridges that are optimized for the instrument, and that produces in our hands pretty superior yields and purities for both IMAC and GST. So just in summary, Profinia is just a nice, easy-to-use device for affinity-tagged purification. It gives the advantage of having that 2D separation profile where you have affinity followed by desalting. You're guided through the methods with the nice user-friendly UI, and we have a whole set of reagents that complement the methods that are found on the Profinia, or found in the Profinia instrument. So, real quickly, let's look at some data customers' labs, and so that's probably more interesting to those of you that are tuning into this webinar. So we've had a Profinia instrument down at UCSD, and several researchers down at UCSD have used the Profinia instrument. So I'm going to show you some data directly from their labs. Dr. Dan Dries, who was at UCSD and he's now doing a postdoc in Texas, wanted to purify a 34 kD GST fusion protein. So he used the preset methods to purify his 34 kD protein, shown in the gel over here in lane 1. He then, after purification, did a cleavage, because typically people use GST tags to fuse a very small peptide onto the large GST moiety. And so in his case, he was interested in isolating an 8 kD target. So he cleaved the protein and then ran it over another preset run and isolated his 8 kD target. And his target came out in the flow-through, and then the GST moiety, which he obviously didn't want, was retained on the cartridge. So this is an example where a small peptide of interest was purified and isolated using the Profinia preset methods with a little customization in between. Maya Kunkel at UCSD has also done some interesting experiments downstream of Profinia. And really the take-home point of this slide is that Profinia allowed Maya to free up her hands so that she can start looking at really what she wanted to examine was protein-protein interaction. So in this case, she isolated a 34 kD GST fusion, and she purified GST as a control, and her GST fusion protein. She then took both of those proteins and probed them on a 96-well dot plot and was able to identify two putative binding partners for her 34 kD protein. So again, the main point from this slide is with Profinia, 10 minutes of her time, the instrument was doing the protein purification. She was there setting up her dot plot, which was much more important than doing affinity purifications via batch or gravity. Satoshi Kajimoto is another researcher at USD who's used Profinia. He was interested in isolating a whole variety of protein kinase Cs. So on the left-hand panel, we see a Kamasi stained gel of 5 different protein kinase Cs that he isolated with Profinia. And he's in particular interested in protein kinase C epsilon. And so here's just a Western blot showing the profile or the fractions that were obtained from a Profinia purification. So here is his specific target protein. That was just another piece of data from the UCSD lab. And finally, in our last slide, this slide really gives an example of where Profinia can be used in custom applications. So in the custom methods, you can basically choose what volume you want to purify, what type of tag you want to use. And in this case, Joel Kim and Susan Taylor collaborated with us here at Bio-Rad, and they sent us some cyclic AMP affinity resin, which we packed in cartridges. And they were able to use those cartridges in the preset method mode, and they were able to purify a cyclic AMP binding protein, actually a truncated form of a protein kinase that was expressed in E. coli. And it's actually expressed at a fairly low level, so they were able to they isolated 300 mils of soup, and they divided it into 2 runs, and they did 2 150 mil purifications on Profinia. So the gel analysis just shows those duplicate runs. Wash 1, wash experiment 1, or cartridge 1, cartridge 2, second wash in cartridge 1, cartridge 2, and then eluted purified protein kinase that was competitively eluted with cyclic AMP. So again, this is a great example where really any type of affinity tag and any type of affinity application can be applied to Profinia. So that's kind of the versatility of the instrument. Any type of on-off purifications can be used. So I know we went through a lot of material, but thanks for participating. And if you have questions, we'll address them now.
So thank you, Laurie. At this point, for those of you who are thinking about questions, go ahead and text them to us. We have a couple of questions that have already been texted, so we'll ask Laurie to go ahead and address those. So CK will show those questions to you. The first one is John asks if the extinction of a protein is necessary to gain the protein concentration in the mouth. We'll let Laurie answer this.
Okay, so it's always good to get questions that I know the answer to. But if I get stumped, I have 2 colleagues here that can help me out. Bottom line is you need to mic on. Do you need to know the extinction coefficient? Well, in general, no. I mean, a standard extinction coefficient, the default is 1. Typically, when you've cloned a protein, and if you've gotten your data from, say, the NCBI database, the extinction coefficient is routinely given as part of the data that's obtained from your sequence. So if you don't know it, you don't have to plug it in. The default is 1, and you'll get a less accurate concentration, but it certainly you don't need to know the exact extinction coefficient. It does help. It'll give you a more accurate assessment of yield and concentration if you do know it. And hopefully that answers your question. Question number 2, fire it at me.
Okay, so question number 2 comes from Sultan. Asks, what is GST and what does IMAC stand for? Laurie?
Okay, so this is a quiz to see if I really know my stuff. GST stands for glutathione S-transferase, and IMAC is immobilized metal affinity chromatography. And for our cartridges, the immobilized metal is actually a nickel ion that's chelated to our Unisphere resin. And nickel will actually form valencies with histidine residue. So that's why for IMAC purification, you typically tag your protein with 6 histidines, and they interact with the metal on the Affinity cartridge. Smaller, it's a 6 amino acid epitope, so that's a little less than 1 kD. GST is actually an enzyme, so it's a 26 kD enzyme that the reason it's used for many applications is if you have a very small peptide that you want to express. Typically, peptides don't express well in E. coli, so if you fuse it to the GST moiety as Affinity-Tagged, you can actually get expression of a small peptide. So GST is glutathione S-transferase, and then the transferase gives you actually insight into the fact that it's actually an enzyme. So GST purification is actually an enzyme substrate type of purification. So hopefully that addresses your question there. So the next question is, how do you know when to change the cartridges? Okay, so here's my answer to that. If you're going to switch to a different protein, I would definitely recommend switching the cartridges. We have obviously cleaning and storage protocols that actually are by default run through the instrument, but you can only get a cartridge cleaned to say 99.8% efficiency, so there's always a chance of carryover. So in general, 1 protein per cartridge. So if you're going to switch to a new protein, switch cartridges. If the cartridges are disposable, they have a disposable frit, so we haven't designed these to withstand hundreds and hundreds of applications. So eventually your cartridge will clog. Clogged cartridges cause back pressure and slower flow rates, and so you want to minimize that happening. So in general, we recommend 5 times so that we can guarantee to you, the user, that you're not going to experience back pressure problems, because that's never fun. So the next question is, can you pack your own resin, such as Protein A? The short answer is yes. The long answer is right now we don't have. So bottom line is, if you can convert, if you pack a column or a cartridge that can be made compatible with lure locks, you can insert a column or cartridge into Profinia, so it accepts any type of lure lock connection. Right now, so that's the answer to that. We, in the future, would like to expand this application to any type of affinity resin, but it's as our we have a marketing person in the room, and as they did help me out here, we do have Protein A cartridges. I don't have a catalog number on that, but we do have Protein A cartridges that are available in the BioScale mini cartridges. So you don't even need to pack your own resin. You can use BioRad Protein A cartridge. And if you don't want to preset the methods yourself, feel free to email and we can get you a protocol for Protein A purification. Can you use your own buffers? Definitely. IMAC buffers and GST solutions are not proprietary, and we're not trying to make, anything more difficult for the user. Some people want to put a protease inhibitor in their lysis buffer and in their wash buffer. Some people want to, if you have a large protein that's really large and it perhaps aggregates with other proteins, some people want to put, say, 10% glycerol in your own buffer. And so you can make your own buffer formulations in the preset methods. You can then plug in the concentration of the buffer that you've made, and also with the instrument, we supply empty bottles, buffer bottles, that can be used to make your own buffer region of the instrument. I was going to show you that slide, but bottom line is, yes, you can make your own buffers. There's the buffer zone. Oops, but now I can't see my questions. Questions now? Can I use your own buffers? Yes.
Earlier, another question was asked by Roger.
I want to know why that question was
what is the cost of the unit, software, buffers, and pre-poured columns? So without having had memorized all the prices and catalog numbers, what we'll request you to do is contact your local sales reps, or even give Bio-Rad a call. So at this point, there are no further questions. This will now conclude today's webinar. For those of you still piped in here, I thank you on behalf of Bio-Rad for your attendance. I hope you're able to gain some insight into Profinia's technology, and that you will find a place for the Profinia system in your workflow. Please have a wonderful day.